Hi. So in previous session we have discussed all about the bacterial infection, and we have learnt the various kind of immune responses developed against intracellular and extracellular bacteria. Then we have looked at how this bacteria is causing disease, and where this disease causing factors or virulence factors are present in the bacterial genome. It could be in the form of extra chromosomal DNA, in the form of plasmids, or it is located, there is an island, there is some chunk of DNA of bacterial chromosome that contain these virulence factor, and all these virulence factor basically triggered by some environmental or external stimuli, the expression of these pathogenicity islands, which is expressing the various virulence factor. Okay, we have learnt about the various kind of toxins, that is exotoxin and endotoxin, and we have looked at how this this microbial pathogen basically trigger the disease when they are infecting the host. So in continuation of this previous session, I will discuss one bacterial disease. This is Mycobacterium tuberculosis, which is a causative agent for the tuberculosis. Okay, so let's begin with tuberculosis. And so tuberculosis is basically caused by Mycobacterium tuberculosis. I will give you some brief introduction about the mycobacteria as such. Okay, so Mycobacterium tuberculosis, we call it as a bacilli. So they are basically rod shape bacteria, and here you can see there is a kind of a schematic of Mycobacterium tuberculosis. They do not form spores. There are some bacteria which makes spores. Okay, one very good example of bacteria which makes a spore is Bacillus anthracis, and due to this property, this Bacillus anthracis can be used as a biochemical weapon. Okay, because spores, you can understand, spores are very resistant entity, and they can be easily transported without any special need. Even in dried form one can transport it, and that makes it that gives the property of a biochemical weapon. Okay, so this mycobacterium is basically resistant to the decolorization by acid alcohol. So what is acid alcohol? It's a mixture of, it's a simple mixture of 95% ethanol, and it contains 3% hydrochloric acid. Okay, so even if you wash, if you stain the Mycobacterium tuberculosis, and if you wash it with this acid alcohol, then it is a resistant to decolorizing by this mixture. Okay, that's why we call it as an acid fast bacilli. Okay, so this mycobacteria, by and large, they are more hydrophobic in nature. They have a variety of lipid and lipid derivative, or lipid and sugar derivatives over the wall. We'll take up this this composition of wall in great detail in subsequent slide in this session. Okay, so they are hydrophobic in nature, and that hydrophobic nature make them very, due to this hydrophobic nature, of this Mycobacterium tuberculosis, this makes a very successful pathogen. Okay, we will discuss in subsequent slide. Okay, so they are resistant to the chemical agent because of hydrophobic nature. Okay, so most of chemical agent are basically hydrophilic in property, like even if you see this this acid alcohol mixture, which is a 95% ethanol and 3% HCl. So due to this hydrophobicity, they cannot decolorize, okay. And they are due to this property, they are resistant to the even drying, okay. This Mycobacterium tuberculosis can survive in dried sputum, okay. So even the sputum is dried, and generally the Mycobacterium tuberculosis, when the infection is too much, then they are present in the sputum, okay. And this sputum, if somebody spit it, and if it is a sun dried, still in that situation this Mycobacterium tuberculosis is alive, okay. When they will get the appropriate host, then this will again this can turn to the virulent, and then that can cause the disease, okay. So this is a generalized property of Mycobacterium, particularly Mycobacterium tuberculosis, and there are various mycobacteria. One is Mycobacterium tuberculosis, which cause tuberculosis, Mycobacterium leprae, which cause leprosy, okay. There is a Mycobacterium avium intracellulae, which basically affect immunocompromised individuals. So immunocompromised individual is it could be a acquired immunodeficiency syndrome people who are infected with HIV, okay, or the individual who has a congenital immunodeficiency. So this Mycobacterium avium can infect, and that can cause the disease, okay. Another is Mycobacterium bovis, which is quite common, okay. So this causes tuberculosis, but this tuberculosis is limited to the cattle, and they do cause in some cases in disease in human, but again the point is that this is basically this cause in immunocompromised individual, okay. There is another group of mycobacteria, which we call it as a non-tuberculous mycobacteria, NTM. So this NTM is basically are bacteria other than Mycobacterium tuberculosis and M. leprae, okay, Mycobacterium leprae, and basically they are present in soil and in another form, and in general they don't cause any disease, okay, but sometimes they cause the disease because of depression of immunity by some or other way. Some or other way means some individual receives some transplant, then their immunity is depressed, okay, or if individual is infected with HIV, okay, so that can also cause the infection of this non-tuberculous mycobacteria infection. So generally this NTM infect the soft tissues, okay. It's not like a classical symptom of pulmonary tuberculosis. It's a to be very important that this infection is very hard to detect, okay. So but they can respond with normal anti-tuberculosis drug, okay, but this is a very non-tuberculosis mycobacteria is not very common. It's a quite uncommon infection, okay. So Mycobacterium tuberculosis, the paradigm for human host pathogen adaptation. So this Mycobacterium tuberculosis is a very smart pathogen, extremely smart. So they will not cause too severe disease immediately, okay, and but they will cause disease very slowly and calmly, and eventually they will affect the host very badly, okay. So overall I want to say that. So Mycobacterium tuberculosis is known to initiate infection most efficiently in lower lung through a small aerosol droplet that contain only 2 or 3 bacteria. So you can understand only few bacteria is sufficient to cause the disease, okay. And Mycobacterium tuberculosis appears to lack the classical virulence factor. So, if you see very carefully, if you see gram positive bacteria, if you see gram negative bacteria, so gram positive bacteria or gram negative bacteria, they have some virulence factor, okay, and this virulence factor basically attract the attention of host immunity, and then host immunity appropriately develop or clear or eliminate those infection through innate immune responses or adaptive immune responses. But Mycobacterium tuberculosis is not like that. They are just sitting silently in the macrophages, and they slowly make their more copies. And if they understand that okay, host immunity is quite aggressive, then they will even go in latent stage, okay, they will not do too much activity, okay. So in general, they don't have a very specific virulence factor against which the host immunity will work and eliminate the infection, okay. They do have, but they do not provoke the host immunity very strongly. That's what I want to say. They use the host macrophages to transverse the host mucosal barrier to sterile site deep in the body, okay. So they basically have a carrier. The carrier is the macrophage. The macrophage is host macrophage. So through this carrier they can move here and there, and they can cause the severe disease. So host immune evasion, modulation and extrapolation are the trump card, okay, of this pathogenic mycobacteria. So Mycobacterium tuberculosis, here there is a little thought, it's a dirty little secret of this Mycobacterium tuberculosis is to be hydrophobic, okay, and fly more efficiently in a tiny droplet to bypass the innate immune system, okay. So since it is a hydrophobic, they are very easily mingle in the system, and then their wall is extremely thick. It's a very resistant overall. So if you see very carefully all over the living world, then you can see that there is a one arthropod which is very successful, which is appeared quite long back, okay, and still it is living very successfully, and that arthropod is cockroach, okay. So cockroach are very efficient and very evolved, okay. If you don't give the food, they will eat plastic. If you don't give oxygen, they will start living without oxygen. So similarly, Mycobacterium tuberculosis is also very successful pathogen, okay, and still they are causing a lot of disease, okay. So how this infection is basically taking place, okay. So tuberculosis we also denote it as a TB, okay. So tuberculosis is a contagious and often this is transported from one individual to another individual through this airborne droplet infection, okay. And it is this tuberculosis is it's not always, it's a little dominant in low income or middle income countries. The number of cases is quite a lot in these countries, okay. TB continued to be a major cause of morbidity and mortality, okay. But this is not fully true, okay. In a developed country also there is a reasonably good number of tuberculosis cases, and over there is some drug resistant tuberculosis are also there, or multiple drug resistant tuberculosis are also there. In subsequent session I will talk about the drug, and at that time I will discuss about the first line of drug, second line of drug, and how to treat the multiple drug resistant tuberculosis, okay. So only active pulmonary tuberculosis is the contagious. Here just I want to say that most of Mycobacterium tuberculosis infect the lungs, okay, and that we call it as a pulmonary tuberculosis. There is another category of tuberculosis which we call it as a extrapulmonary tuberculosis. Okay. So basically this Mycobacterium tuberculosis infect the macrophages. Okay, and these macrophages are mainly the first infection is mainly in the lungs. Okay, through respiratory route. But as you know that the macrophages are present all over our body. Okay, in bone there are macrophages, in connective tissues macrophages are there, in liver, in another almost all tissues has a tissue specific macrophages. Okay, so these Mycobacterium tuberculosis can infect those macrophages, and whatever infection caused by Mycobacterium tuberculosis other than lung, we call it as a extrapulmonary tuberculosis. For example, the Mycobacterium tuberculosis is infecting the bone macrophages, okay, which you call it as osteoclast. Okay, so if it is infecting, then that will cause the bone tuberculosis. Okay, and it is please note the pulmonary tuberculosis is reasonably very easy to manage, but extrapulmonary tuberculosis, it's extremely difficult to manage. Even the diagnosis is very difficult. Okay, finding that okay the bone has a tuberculosis, it's extremely difficult. Generally people confuse with bone cancer or other diseases. So the extrapulmonary tuberculosis is extremely difficult cases. Okay. So now I just want to take to the history, because I have discussed about the cockroach, and I have discussed how Mycobacteria is very successful, and we have all evidences. Okay. So tuberculosis probably occur in as an endemic disease among animal long before it affected human, and from the time of Hippocrates, tuberculosis was known as phthisis. Okay, it's a term derived from Greek, means wasting away. Wasting away means the healthy individual started losing their weight and energy level and everything. Okay, so this we call it as a wasting condition. Okay, losing weight. Okay, getting more thin and like that. So this we call it as a wasting away. Okay. So existence of tuberculosis in mummies dating 2000 to 4000 BC. So it is already very well documented and reported that such a old mummies they also has a tuberculosis. As here you can see there is a one Egyptian mummy, and this mummy was having a tuberculosis which is verified by the PCR. Okay. Here you can see the lesion of tuberculosis on the spine of Neolithic man. It's extremely old, right? And here you can see that tuberculosis decay has been found in a spine of Egyptian mummy. Okay, so this is very clearly demonstrate that this pathogen is present from quite early time. Okay, so many fundamental concepts of biology emerged in the context of inquiry about the nature of tuberculosis. Okay, and basically this tuberculosis was discovered by Robert Koch in 1882. Okay, as a causative agent of tuberculosis by an airborne infectious disease caused by organism of Mycobacterium tuberculosis. Okay, and at that time it was this tuberculosis disease was known as a white plague. Okay, because it leaves the white lesion in the lung. Okay, this is a global incidence of active tuberculosis. Here you can see that our country has in India there is a lot of tuberculosis cases, and if you see carefully, then you can find out that this tuberculosis, the number of tuberculosis cases is not basically depend on the climatic condition. Okay, here you can see that close to the North Pole also there is a lot of tuberculosis cases. If you go to the South Pole also, there is a reasonably good number of tuberculosis cases. And of course, this Asia continent, including India, this has a lot of tuberculosis cases. Okay. Now let's look at this, how this Mycobacterium tuberculosis, the wall and the other things are there. Okay. So this is a cell envelope of Mycobacterium tuberculosis. Here you can see that the cell wall is basically, or envelope is basically divided into several distinct groups. Okay. One is that mycobacterial outer membrane. So this mycobacterial outer membrane basically resemble with gram-negative bacteria. Okay. So gram-negative bacteria do have an extracellular membrane. Okay, and that contain the LPS. Okay. So similarly, the Mycobacterium tuberculosis have a mycobacterial outer membrane which contain, here you can see that which contain mycolic acid. Okay. There is a diacyl trehalose. Here you can see that there is a sugar derivative is there. There is a thiocurol dimycoserate. There is a, this is a quite complex molecule. There is a polyacyl trehalose. Okay, and there is a sulfoglycolipids. So these are very complex molecule, and if you see very carefully, it's basically the more, if you see carefully, then you can find out that this is more lipid and lipid sugar derivatives, and long chain carbon compounds are there. Okay. Below this mycobacterial outer membrane, there is a AGP, which is a stand for arabinogalactane peptidoglycan. So again, there outside this in trace of gram-negative bacteria, out there is a outer membrane, and below that there is a thin peptidoglycan is there. So similarly, something similar present in the Mycobacterium tuberculosis as well. And this is basically AGP, arabinogalactane peptidoglycan, which is consist of galactosamine residue, arabinogalactan and peptidoglycan. Okay. So, and here you can see the image that it is quite complex. Okay. Below this AGP, there is a periplasm, and this periplasm is again containing the complex lipid sugar molecules. Here you can see that there is a LAM, lipoarabinomannan. There is a LM, this is lipomannan. So again, this is a derivative of sugar and lipid. Okay. And below periplasm, there is a MIM. This is mycobacterial inner membrane, and this is consist of phosphatidyl ethanolamine. Okay. And phosphatidyl ionositol, and DPG, this is a diphosphatidyl glycerol, and there is a tri or tetraacetyl phosphatidyl ionositol dimanosides. Okay. And there is also tri and tetraacetyl acyl phosphatidyl ionositol hexamonoside. So this you can understand, this is a quite complex complex molecule which is made from sugar as well as lipid. Okay. Now, what is the function of this this thick envelope? Okay. So mycobacterial cell wall induces delayed type of hypersensitivity. So this if it is injected into the animal or human, then it will induce the delayed type of hypersensitivity, and due to this property, it is used as a complete fluent adjuvant. So if you remember, the complete fluent adjuvant is basically consists of heat killed dried Mycobacterium tuberculosis mixed in the mineral oil. Okay. So mineral oil is basically a oil kind of thing. In that there is a dried powder of Mycobacterium tuberculosis we add, and this component basically induces the delayed type of hypersensitivity. So delayed type of hypersensitivity will enhance the immune response. Okay, so whatever antigen is present in this fluent adjuvant, then there will be a slow release, and then there will be a delayed type of hypersensitivity reaction. Okay. So, unlike most gram-positive bacteria, these bacilli augment their peptidoglycan wall with extensive array of complex lipoglycan, which reflect the necessity to resist desiccation in soil environment. Okay, I have explained, I have told you in previous slide that even this bacteria will survive even if it is dried, even it is sun dried. Okay, why? Because of this property. Okay, the thick waxy cell wall or ability to catabolize fatty acid. Okay, so this has a lot of fatty acid. Okay, seems to be useful pre-adaptation to a parasitic existence. Okay. So, with this I will stop the first session about the Mycobacterium tuberculosis. In next session I will discuss about the pathology and test, and then subsequently I will discuss about the anti-tuberculosis drugs. Thank you.